Liquid connective tissues like blood and lymph are essential, carrying nutrients and oxygen and protecting us from disease. Hello, wonderful attendees. I'm your host, Dalvinder Kaur from Lovely Professional University. Get ready to dive into today's topic that is all about your liquid connective tissues. Before we proceed further, allow me to introduce the experts of that day. Dr. A. Najita Banu, Assistant Professor, Department of Zoology, School of Bioengineering and Biosciences, Lovely Professional University. Dr. Banu holds an impressive academic background with an MSc, MPhil, and PhD in zoology from Tamil Nadu. With over 7 years of research experience, Dr. Banu has made significant contributions to the field of agriculture and medical entomology. She has an extensive publication record with 40 research papers to her credit. So I would like to introduce the admission nominee also in this webinar, Dr. Tushchandar Preet Kaur, a distinguished professor at Lovely Professional University, Mittal School of Business. With over 15 years of academic experience, she holds a doctorate in economics, MPhil, and UGC NET qualification. Dr. Kaur is also an author and served as a resource person for seminars and conferences. Before we proceed further in today's session, I would like to request everyone for any questions. You can write in the chat box or in the Q&A box, and same will be addressed from my side in the end of this webinar. So now I hand over the things to Dr. A. Najita, and it would be an absolute delight to listen from her. Over to you, Dr. Najita.
Thank you, ma'am. Thank you for the introduction. Yeah, it's me, Dr. Najita from Zoology Department. I'm connecting with the Lovely Professional University last 8 years. Okay. So today I'm going to be handle the session, particularly all about our liquid connective tissues. Okay. So let me first share my screen, then I'll start the sessions. Okay. Yes. So, our human vascular system, if you're going to be connected with our vessels, okay, vessels I'm talking about are blood capillaries and then veins, it approximately 70,000 miles. So you can able to go up from New York to San Francisco about 24 times. Okay. So here you can able to see the blood vessels. The blood vessels, particularly here, we are going to be seeing liquid connective tissues, right? So liquid connective tissues, we can call it as a fluid tissues. Okay, the fluid tissues, it is very essential for our survival. Okay, so there are two portions in our liquid connective tissues, basically blood and the lymph. So the blood, if you're going to be see, the blood, it is a formed elements. What I'm trying to mean, what is the meaning of formed elements? So if you're going to see only blood, it's not a blood cells. So it is containing plasma as well. Okay, here you can see in the PPT. So the plasma, it is a major portion. The plasma actually fluid in nature. But the RBCs, that is erythrocyte, or otherwise we can call it is a red blood corpuscles. So the red blood corpuscle, it is a formed elements. So it is only a living part of our blood. Okay. So the liquid portion is plasma. So what is the essential role of the blood? Actually, the blood, it does very, very important functions it is playing in our everybody body. So the blood, it is first of all, it is like you can see the oxygen transformation from the external environment to your internal environment. It means inside your body, and then maintaining your body temperature, as well as you can able to see it will protect from your any kind of the enemies means foreign particles and pathogens. So the plasma, it is a liquid portion. The plasma, what is the role of the plasma then? So actually plasma contains a very, very important things, that is a coagulant, anticoagulant. Simply the coagulant, it is a fibrin. The fibrin normally in our body, it is in a simple form of the fibrinogen. Okay, it is an inactive form. The inactive form, when if you're going to be get any damage in your blood vessels, the fibrin, with the help of the clotting factors, it will be converted into the fibrin. So that fibrin immediately clotting your blood. If it is not clotting the blood, what happened? The blood it is going to be leak out from our body continuously, it is cause a lethal effect. It means fatal effect to the particular individual. Okay, the formed elements, not only the RBCs, you can see the formed elements, or otherwise we can call it as a living cells. Okay, the living cells RBC, WBC, as well as you can able to see platelets. So RBC, simple, it is look like a simple a donut-like structures. You might be aware about the donuts, or otherwise we can call in South medu vada, right? So the donut-like structure, and then WBC. So the WBC means white blood corpuscles. The white blood corpuscle, it is playing the very important role to maintain your immune system. There are two main categories in the WBCs. What are the two main categories? One is you can able to see granulocyte, another one is a granulocyte. Here you can able to see some of the images look like a purple color. Okay, the inside you can see this is a nucleus part. Okay, the outer surfaces we call it as a cytoplasm. The cytoplasm, some of the cells are, it is a some minute granules are present, but some cells here you can see without granules. So the two main category, it is a granulocytes and agranulocytes. So the granulocytes are three cells are coming in at the granulocytes. So they are neutrophil, eosinophil, and then basophil. Your lymphocytes and monocytes are agranulocytes. So each and every white blood corpuscles are playing the different roles in your body. But basically, the all are ultimately maintaining your immune system. It is fight against the foreign particles. Okay. So this is a composition of blood when we are going to be take the, for example, any kind of the hematological experiment. What is hematology? Hematology, it is nothing to study the bloods. Okay. So the hematological experiment, usually you might be seen in the doctors, they used to take with the help of syringe 5 ml of blood. Okay. For any kind of you have allergic infection, any other infection, maybe a viral infection. Okay. So then after that, if you're going to be, you want to see what is a plasma and then what is a formed element, you can able to simply keep that particular blood after it's transferring into the test tube. If you are keeping as it is in the laboratory level or in a room temperature, you can see two different portions are separated. It means it is automatically naturally having the ability to coagulation because the fibrin is present. Okay, now look at, see, in the 5 ml of blood, if you are going to be observed, the portion which is settled, we call it as a pellet portion, the settled one or coagulated one, that is your formed elements are living portions. The upper portions are fully liquid in nature. That liquid is your plasma. The plasma, 55% of in your blood, it is usually plasma only. So rest of the 45% are your formed elements are living cells. In the plasma, 91% are water and then 7% of blood protein. The proteins are, you can say, albumin, globulin, and then fibrinogen. The 2% are nutrients like amino acids, sugars, and you can able to see the 1%, very least amounts are electrolytes, and then hormones are present in the plasma. Okay. Then in the cellular component, I already explained the cellular component, your RBC, WBC, and then platelets are present. Okay. Now come to the next one. As I already discussed here, you can be able to see the donut-like structure. This is a shape of your RBC. Okay, actually, where the RBC, the blood cells are originated. Actually, the blood cells are originated from, you can able to see the bone marrow. Okay, the bone marrow, very, very important. Actually, you may be studied about the stem cells, right? So the bone marrow, it is act like a stem cells. Actually, the generation, synthesis, formation of your all cells in your body, it will be originated from your bone marrow one. Okay, so simply donut, or otherwise you can call it as a biconcave like structures. The average lifespan of RBC, it is 120 days. Okay, so it means it is cyclically, it will be go once after 120 days, it will be destroyed. Okay, in the spleen, usually it is going to be destroyed. Okay, so the hemoglobin, it is very, very important factor which is present in your RBCs. So it is iron-rich protein molecule. Okay, so you may be come across this word hemoglobin. The hemoglobin level is low. It means a condition of anemia, right? Now, here I'm not going to be theoretically, I don't want to bore you. So today we will see some like an interesting part. We will see like a live example how we can able to count your RBC, total RBC. Okay, now this is a flow diagram which is indicating the where actually RBCs are formed. The first one, it is a hematopoietic stem cells. So we call it as a shortly HSC. The hematopoietic stem cells are pluripotent stem cells. What is the meaning of pluripotent stem cell? Pluripotency. Pluripotency means it can able to form so many different type of the cells. Okay. Then multipotent. Multipotent means it is a subset of cells. Okay. And then from the hematopoietic stem cell to multipotent progenitor stem cells are originated. From this multipotent progenitor stem cells, it will be gives rise to the megakaryocytes, granulo, mega progenitor cells, and then your lymphoid progenitor cells. Okay, so the erythrocytes are formed. Erythroblast is simply we call from the erythroblast your erythrocyte, that is your RBCs are formed. Okay, now so the erythrocytes normally in normal healthy human being, if it is a male, means 14 to 18 grams per 100 ml of blood. Or otherwise, if I'm going to talk about in number, if you're going to be seeing number, it is a 4.8 to 5.4 in case of male as well as in the female. It is an average, okay, per microliter, okay, per microliter of blood containing 4.8 to 5.2 RBCs. In case of female, 12 to 16 grams, okay, in 100 ml of blood, okay. If any kind of abnormalities, so as I already said, if it is a condition, your hemoglobin level is low, so that is a condition of anemia. Okay, that time you feel some kind of fatigue situations, some kind of weakness, maybe your eyes and then your skin colors are little dull, look like a dull skin color. Okay, as well as sometimes you feel some breathing issues also. For example, you are working continuously, okay, but you are not taking the breakfast. Okay, so after sometimes you can automatically feel some weakness, as well as you might be feel the breathing issues also due to the your oxygen not getting proper to the your hemoglobin not able to uptake of the oxygen properly, because hemoglobins, the structure of the hemoglobin, if you are going to see, it is a 4 oxygen binding sites are present. Okay, it is helping to transport the oxygen to your tissues. From the blood cell to tissues, the oxygens are simply transported. Okay, now this is about some basic informations about RBCs. Okay, your red blood cell. Now we will start how to count the total RBC which is present in your blood. Yes, you can see some of the checked kind of diagrams. So actually it is nothing. Here it is a one instrument we used to call, the name of the instrument, that is your new bar counting chamber. So instead of showing the diagrams, I'll go for the live session so you can able to get the information more crisp and clear. Okay. And now I'm going to be show these are the instruments. Give me 1 minute. Yes. Okay. So now you can able to see this is a hemocytometer box. Here it is mentioned. It is a hemocytometer box. The hemocytometer box, or otherwise we call it as a new bar counting chamber. Okay.
Hemocytometer. Interruption, Dr. Najita. Can you please stop sharing your screen?
Okay. Okay.
Yes, you can continue now.
Yes, yes. Now you can see it is a new bar counting chamber. Here it is mentioned clearly new bar counting chamber. Okay, new bar, why this particular instrument it is called like a new bar counting chamber? Because the scientist who is invented first time this simple like a compact instrument, that's why we call it as a new bar counting chamber, right? Now you can see in the new bar counting chamber, it's look like a letter H. So there are two depression in this structure, right? As well as there is a two partition here you can able to see. In that partition, the square-like structure which is you observed in the like a slide, it is present here. Actually, it is a microscopic in nature, that square, so we are not able to see in the naked eye. So I will show you that particular microscopic structure of that particular a squares also. So using this new bar counting chamber, not only the RBC, we can able to measure a total WBC, that is your white blood corpuscle counts also. Okay. So for that, we need some other instruments, accessory instruments. What are the accessory things we need? Now you can able to see this is a pipette. Okay. This pipette we call Toba pipette. Okay, Toba. So again, the scientist who is invented this pipette, the name of the scientist is Toba. That's why we call it as a Toba pipette, or otherwise you can call it is a RBC pipette. Why we call RBC? There are two pipettes are there, right? So here you can able to see the bulb is present inside the bulb. some small red color beads are visible to you? Yes, everyone. So these beads are look like a red in color. If it is look like a red in color, that pipette usually used for RBC count. Okay. If the beads color is containing white color, that pipette usually used for your WBC count. Then after that, you can able to see there are two readings are here. Okay, like a 0.5 and then 5 and then 101. Now what we are going to do, so this is a structure of your pipette, right? I will tell you how to demonstrate. I am going to be demonstrate. Okay, so this is a nature of your pipette. Then you can able to see after that whatever the things we need a lancet. Okay, the lancet, it is a, see, this is a small lancet. It's a kind of strip only. The lancet, it is a only one time usable. It is made up of stainless steel. Now you see, you have to open like this. Okay. Now you can see the stainless steel lancet. Okay. So in the lancet, the corner of the lancet, you can be able to see it is a very sharp pin is there. Okay. It is only one time usable. Once you are breaking your fingers, after that you have to dispose this lancet, okay, properly. Now, just I'm keeping now, then I'll, once I'm at the time of demonstrating, I will use the same lancet. Then you need this dilution fluid. Okay. So this dilution fluid, we call it is a RBC diluting fluid, or otherwise we call it is a heme diluting fluid. Okay, this fluid, it is commercially available, as well as I'm having that like a description. If you want detailed composition of this fluid, I will share you that detailed composition of fluid also. Okay, now what we are going to do, first we are going to be break our fingers. Okay, let's start. Before breaking our fingers, first we have to surface sterile our finger. Usually we are going to be use our ring finger. Okay, so you can see the ring finger only going to be used for the breaking your like a for your RBC. So now I'm going to be surface sterile or like a finger. See, this is a ethanol. The purpose I'm going to be used for sterilization. Okay. Now you see, I'm going to be sterile my finger. Okay. First, you have to surface sterile your finger. Once sterilization is over, just let it be dry. You can use otherwise cotton wipes also. Once it is done, first you have to give a gentle press on your fingers, because the blood flow, it is continuously going on. When you are going to be massage your finger, you will get the more flow frequent. Okay. Now what I'm going to do, you can see. So now I'm going to be used lancet. Okay, this lancet. Okay, using the lancet, I'm going to be break my finger. After breaking finger, once the blood it is coming out, you have to take the blood up to the reading of 0.5. Okay, up to the reading of 0.5, you have to take the blood with the help of your that RBC pipette. Okay, if you want to go for the WBC count, you can use that WBC pipette, which is containing the particularly white like a beads. Okay, now I'm going to count only RBC. That's why I'm using the RBC pipette. Okay, now you can see. After that, I'm going to take some of the heme fluid also, because the heme fluid you have to take up to the reading of 101. Okay, once you are taking the heme fluid, then you have to go for the hemolysis. What is a hemolysis? You are going to be lyse your particularly RBC with the help of your heme like a heme fluid. Okay, so heme diluting fluid. I'm going to be dilute my blood. If you're not going to be dilute your fluid with the or otherwise hemolysis process, your blood going to be get coagulated, because the fibrin is present. Once the blood it is coming out, and naturally the coagulation factor is present in your blood, it is immediately coagulated. Okay, so once you are taking, you have to go for the proper vigorous mixing. The mixing, not in the vigorous mixing means not like this. Okay, you have to take your in the horizontal portion, you have to rotate smoothly. Okay, likewise. Once you rotated the buffer is your heme fluid as well as your blood. It will be get mixed properly. Okay, after it is mixing, then you are going to be load the particular fluid in between this depression which is present in your hip, this area. Okay, this area. Before that, I cover this area with the help of the cover slip. Okay, because the liquid portion, it is not going to be loose out from this area. Okay, once the fluid it is loaded, leave for 1 or 2 minutes. then go and observe under the microscope. The cells are very clearly visible. Okay, so I will demonstrate, okay, one by one. So my finger is already surface sterile, so that's why I'm directly going to break my fingers and then I will take the solutions. Now you can see the blood is coming out, right? So I have to take the blood from the reading up to you can see up to 0.1, right? So up to 0.5. Now I'm going to be loaded with the heme fluid. Okay, heme fluid. So the heme fluid you can able to see. You have to take in a 101. So simply you have to just circle. Now you can see the fluid is reached into the 101. Now I'm going to be mixed. Okay. If you're not mixing, it will be get clotted. So this is a process we call hemolysis. Okay. The process we call it is a hemolysis. So it will be 1 or 2 minutes. You just mix the buffer as well as the solution going to be mixed properly. Go for the gentle mix continuously for 2 minutes. Okay. Now you take the cover slip. Okay. See, this is a thin slide-like structure. The slide-like structure we call it as a cover slip. You just keep the cover slip in your newborn counting chamber, any one side. You can use this side as well as this side because the grid it is present in a both side. Okay, you can use any one direction. You can use this side or otherwise this side. Now I'm going to be load the sample. Actually, your hemocytometer or otherwise your newborn counting chamber having the depression here, you can able to see it's look like a canal-like structure. So it is giving the channel to flow your the particularly mix. Okay, now I totally loaded the sample. Okay, now we are going to be observed under the microscope. I think everybody you are ready. So I think you are curious about seeing your my RBC, right? Let's see. See here, you can able to see this is our compound microscope. In the compound microscope, see, it is called like a binocular microscope. Okay, it is a compound but binocular. Why we call it is a binocular microscope? Because you can see there are two eyepieces are there, so we call it as a binocular microscope. So you can see your RBC, it is a diameter is 7 to 8 micrometer in diameter. So you can able to observe in the low magnification. See, low magnification means 10x. Okay, in the 10x you can see it is a nose piece. In the nose piece you have that objective lens. Through the objective lens you can able to observe very easily. Now you can see I already fixed the like your newborn counting chamber. See, this is a stage. In the stage we have a clip for the supporting. Now I fixed our newborn counting chamber. Now it is fixed. Now you can able to check the yeah. Now light is on. Now just I'm focusing. Yes, it's very beautiful. So you can see through the lens. I'm using my camera. I will show you through the lens how it is look like your RBC. Look at the structure. I think so. It is clear, right? Yeah, the cells are, see the dotted one, you can see the small dots. The dots, it is your RBCs. Okay. So now what you're going to do, you have to count all the cells. Yes, you're hearing properly. I think you have to count the cells because it is taking a time-taking process. you have to count not only this single squares which is you're observing in the microscope under my camera. Okay, actually you have to count more than 4 more squares are there. You have to count all the cells. Then you having some equation how to exactly calculate how much number of cells are present in your blood. Okay, I think it is clearly visible. Now let's rinse. Now I'm going to share with me rest of the information you can able to see here. The theoretical information, which is your observed. Now you see. So whatever the things you observe, like in the hemocytometer, now you see the small square which is you observed in the laboratory. The H1 I discussed with you. Yes. Now look at the hemocytometer Newbar counting chamber. The Newbar counting chamber, here you can see in the 4 corners are there. In the 4 corner, that is usually used for the WBC, that means white blood cells counting. Okay, so the depth of the you can able to, the squares are 1 micrometer. So the 4 corners are usually used for the WBC count, but today what we see in under the microscope, there is a center square. So you can see the center one. In the center one, I only focus in a 1 corner. So in the 1 corner, if you are going to be observed, it is how many number of cells are present? 1, 2, 3, 4, 5. 5, as well as internally also there are 5 cells. Look at in this next image. Now I think here it is clear. Now you see this 1 corner only I focus with you. So we have to count 1, 2, 3, 4 and then center one. Is it clear everyone? So 1, 2, 3, 4 and then center one is 5. We have to count the number of cells which is present in the each individual cubics. Okay, after that only you can able to see how many numbers of RBCs are present in your body. So these are the information which is I already discussed when I was performing the experiment. Okay, the 0.5 mark of the RBC pipette. Then you have to use the heme solution, that is your isotonic solution, up to 0.1 mark. Then we mixing vigorously for the hemolysis purpose. So as I already discussed with you, I will give you the detailed information about the RBC diluting fluid solution or heme fluid solution. So here you can able to see it is containing the mercury chloride, sodium sulfate, and then distilled water. It is commercially available. There is no need for the preparation. If you are interested to perform, because if your work is related to hematological work, if you are doing PhD in the hematological work, then you can prepare, you can store it for the longer time period. Okay, so the same, like you can see the squares which is I already discussed with you. Here it is mentioned W for your the white blood cells, and then R for it is a red blood cells, RBC. So the Newbar counting chamber procedure. Okay, now under the microscope you have placed your like a Newbar counting chamber. Then we have some procedure how to count the cells. Now look at the structure, the cells which is present in the line. Okay, this line, if the cells are 70% are touch in yeah, only in the squares only we consider for the counting. Okay, the counting method, we have to count the each cell in present inside the square zigzag manner. Okay, each square is zigzag. Now you see 1, 2, 3, 4. For example, in the first square, so these cells are 70% are occupied. So I'm not going to be considered this one, but I can consider this one. So number of cells present at the RBC present in the square one is 1. The second one is 2. So likewise, you have to count first row, then you have to come down, then go for the zigzag manner in the second, third, 4. Okay, so it will be go like that. Likewise, all 4 corners you have to count all the RBCs. Once your counting is over, now I fix the magnification in a 10x. Okay, sometimes some students are not able to see in the 10x magnification clearly. Okay, so that time you can go for the 40x. 40x means the size of the cell, it will be magnified. Okay, see, this is a one which is we observed under the microscope. Okay, the cells are look like this one. Okay, now see, so this corner you have to count. Let's see here, the 3 lining, it is indicating you have to count only in the cells which is present this particularly inside the 3 line large squares only. Okay, so it means 1, 2, 3, 4. Then come down 1, 2, 3, 4. Then come down 1, 2, 3, 4, and come down 1, 2, 3, 4. Okay, so likewise you have to count. Once your counting is over, now you see this is a calculation we are going to be used how to count how many numbers of RBCs are present in a 1 microliter. I am talking about a single 1 microliter of your blood. Okay, so this is a method. Now this formula, if you're thinking, ma'am, this formula is not that much real, so you can go with this formula also. This is the simplest one. Okay, so number of RBC per cubic mm of your whole blood, it means number of RBC counted. It means you are counted the individual cells, and then dilution. The dilution according to the like a squares, the dilution is 20. We consider it as a 20. Okay. Then area counted, how many squares you are counted, and the depth of the fluid. Okay, this is the formula. Now you see here the dilution is 20. The area counted totally we are considered 5 like a squares. The top 2 corners and bottom 2 corner and then center squares one. Okay, top 2 corner, and I'll show you now. Then you will get the clarity. See here, you see 1, 2, 3, 4. Top 2 corner and bottom 2 corner and then center one. Okay, that's why we here we mentioned it is a 5. The number is 5. Okay, then after that, this is the depth of your square 0.04. Then it is 0.0 square meter. The depth of the fluid 0.1. The finally the number of RBC in your whole blood you can count through this smallest simplest formula. Okay, after your counting you will get the value of 4.7. Sometimes it is not exactly 4.82, 5.2 you are going to be get. So sometimes you can able to see it will be 4.5 or 4.7. If it is going below that, below in the sense below like a 4 million, okay, below 4 million, like a 3.5, that is a condition of the anemia. Okay, but sometimes the cells are produced more in number, more than 5.4. So what is the condition? See, actually, if it is a more 0.5, it is a condition of polycythemia. Okay, what is polycythemia? The polycythemia vera, it is a some difficult condition. Why this polycythemia disease usually occurs in the human beings? Because the mutation in JK2 genes. Okay, the gene particularly in your DNA, the gene get mutated, so it will be going to be directly target your bone marrow, because I already discussed with you in the bone marrow only where the RBCs are produced, right? RBC synthesis takes place. So continuously bone marrows are started to produce more number of RBCs. Okay, so that time, so the number of RBC counts are more in the particular patients. Okay, so that condition, the polycythemia, it is, you can call it is a cancer. It is nothing, it is a cancer, but the cancer because of your RBCs, that's the reason why we consider it as a polycythemia. Okay, so fully we call it as a polycythemia vera. Polycythemia vera, usually it will be target your bone marrow. In the bone marrow, specifically in your DNA, specifically your JAK2 genes. Okay, JAK2 genes. Then, but it is a critical condition. Okay, but when the ma'am, the number is, it is a 4.1, that is also polycythemia condition. No, it's not like that. So that condition we call it as a anemia. Okay, anemic condition if the number of cells are less, but number of cells are more, that is a polycythemia. Okay, if it is going for 6,000, 6 millions and then 7 million, that condition is only polycythemia. Okay, so this is all about from my webinars. So here you can able to see this is my backbones. Okay, they are my backbones. Here you can able to see my BSc, MSc first year, MSc second year, and PhD. Okay, the students are like a collectively working. They publish the paper in a 4.5 impact factor. I'm talking about, yes, you are hearing properly. UG student. See, the corner one, he's a Rudradev, Akshat, you can say Ritu. My backbone, they are supported me to like a glow in the area of the research. Okay, so that's all from my side. Thank you for this listening patiently my lectures. So if you have any queries, you can ask me. Dear participants, I think I'm audible to you. If you have any query, you can discuss with me.
Thank you so much, Dr. Najita. So it was wonderful watching you talking on all about your liquid connective tissues in depth. So I would request to the admission nominee, Dr. Tashinder Preet Kaur, to be on the screen. Over to you, Dr. Tashinder Preet.
Hi, Madam Dalwinder. How are you?
I'm good, ma'am. Thanks for asking.
Yeah. Is my voice audible to you?
Yes, you are audible.
I don't have screen sharing rights. Can you please give me screen sharing rights?
Dr. Najita, ma'am, can you please stop sharing your screen? Now, Dr. Tashinder, you can share your screen now.
Just want to reassure that my screen is visible.
Yes, Dr. Tashinder, your screen is visible. You can continue.
Thank you so much. So, very good afternoon to one and all. I, Tashinder Preet Kaur, would be briefing you regarding what are the key points which you should keep into your mind before identifying a place to join in. I'll brief you regarding what all accreditations the university has, the kind of exposure the university give to the students. So, by not taking much time, allow me to share the information which I have with me. The university proudly receives highest NAC A++ score from the NAC agency with the 3.68 score on 4-point scale, which is the highest scale in first cycle of accreditation among all government and the private universities. Continuing with, apart from NAC, we have ACDSP. We have certain approvals from UGC, from UGC Distance Education, National Council for Teachers Education, Council of Architecture, Bar Council of India, Punjab State Council for Agriculture Education. The list is long. This is just a glimpse. So we do have certain memberships as well. Moving ahead with the rankings which university has, the university has been ranked second by World University Rankings. This is the first attempt the university has secured global brand for 101 to top 200 universities. From the Atal Ranking Association achievements, we have been ranked third because of the innovations the university is having. This gives me immense pleasure to share that the university, the students not only showcase their performance, their talent in Tokyo Olympics, rather they also created the history not only by participating but winning various medals in the Tokyo Olympics. And they not only made us proud but the whole country too. So the face is not new to all. This is the one of Neeraj Chopra. We are well versed what he brought laurels to not only to university but to across the globe. Yeah, continuing with the same, this is a kind of exposure which we provide to our students. Now, the university proudly hosted 106th Indian Science Congress, which was inaugurated by Honorable Prime Minister Shri Narendra Modi ji, where he renamed the slogan Jai Jawan, Jai Kisan, Jai Vigyan, Jai Anusandhan. So, specifying or putting a special emphasis on research. This name is, and this phase is also not new to us. It is he's Nobel laureate for peace and great spiritual leader from Tibet, Dalai Lama ji. He graced the convocation at LPU. This is a glimpse where you can see the two presidents of Asia's great nation gracing the convocation at LPU. He is Union Finance Corporate Minister late Shri Arun Jaitley ji, who inaugurated the startup school at Lovely Professional University. Former Vice President for India also graced the convocation and gave the various inputs to the students to lead a good life. He's Gorgopal Das ji, life coach, style, and the motivational speaker, interacted with the students and guided them the right way to choose the various pathways of life. So apart from the great leaders, the university has welcomed various celebrities, Shah Rukh Khan and Deepika Padukone, the glimpse of whom you can see. He's former captain Mahendra Singh Dhoni. He also visited us and interacted with the students. So you can see the fun and frolic which the students have. This is a glimpse where we got Guinness Book of World Record for the largest Bhangra dance at LPU. So now I'm going to initiate with what all infrastructure facilities the university has. The university has amphitheaters which are used to organize nukkad nataks, events, etc. This is a glimpse of one of the largest stadium, Shanti Devi Mittal Auditorium, which is used to organize the events of national and international repute. Um, this is the academic infrastructure which we have, the Mac labs, the libraries, multi-storied libraries are there. This is the innovation studio which you might have heard about, the uni center which is run by the students, student entrepreneurship, small ventures they take. Allow me to take you to inside. Yeah, this is the inner view of Unimall, where you will find a multi-storied building with all the basic and things you can have, from stationery to the sweet shop, and there is no need to go out, so you will be getting every single facility within the periphery. So the inner view of those who are gym freak and they are fitness, they are oriented towards the fitness. For them, we have the gym facility. To bring you out from the academic monotony, we have the bowling alley. So we are worried about your health. So if you fell sick, God forbidden, so we have medical fitness for you. We have the 24/7 medical facilities for you, and we do have certain tie-ups with the hospitals outside the university. So this is the day view of the university. This is the view of the lush green gardens. Continuing with the facilities, uh, the night view of the university in general and the residential facility in particular. So this is the Shanti Devi Mittal Indoor Stadium with multifarious facilities. I'm taking you inside. Just have a glimpse. Badminton courts. You can find the students in the evening time. They spend their time by playing and opting for any game. And going ahead with this is a shooting range, basketball, lawn tennis, the swimming pool. We have 3 swimming pools. All weather swimming pools are there. So this is all about, this is just a glimpse which I showcased to you. Now I'll take you to the website where I'll brief you. I really want you to have pen and page with you so that you can write the eligibility, before that the duration, eligibility, what all fee structure, and yes, how I'll tell you the ways how you can save your money. So I'm just taking you to the website. All right, now you can see on the screen that I have logged in into website. It is lpu.in, wherein we have long more than 250 programs. Now looking on to your orientation, I have selected biotechnology. So BSc biotechnology is a 3 years program. If I talk about honors, BSc honors biotechnology, it is a 4 years program. So looking on to the eligibility, BSc Biotechnology, it is 60% in 10+2 with the combination of English, physics, chemistry, maths, bio or biotechnology. And continuing with the fee structure. So these are the, it means the person who is scoring 60% in 10+2 can directly go ahead with admission. Going ahead with the fee structure as well. I'll just give me a minute. Just 1 second. Yeah, just sharing my slide again. Yeah. Okay. The fee for this program is 80,000 per semester, and in this program. There are 3 slabs: 28%, 33%, and 38%. We do acknowledge your hard efforts, what you have done in 10+2. And if you plan to appear in university's exam, LPU Nest, that is a test which serves two purposes: eligibility and scholarship. The test is not required for the eligibility, but yes, you can appear for the scholarship. So 28, 33, 38 depends upon either previous qualification or the university's exam. So better you score, better will be the scholarship. So apart from the tuition fee, we have the examination fee, that is 4,500 per semester. So continuing with this, I'll take you to the point where what all are the important dates. The last date to take admission is 31st of May. Today is 30th. Don't wait for tomorrow to book your seat, because any important movement left, you may face the certain reduction in the scholarship slab as well. So don't wait for the last date. You need to sign up. You need to join at sign up at lpu.in. You need to fill the application form. You need to either apply LPU Nest, and then you can directly plan ahead with, or if you want to skip LPU Nest, because there's a time, this is 31st of May is tomorrow. So you book your admission by paying the provisional amount, and then you can apply for LPU Nest for the better scholarship, because LPU Nest is not mandate. So if any case you have any kind of query, feel free to connect us by scheduling a call, by just doing WhatsApp to us, by live video counseling, or you can find out the LPU in your town. So these are the ways through which you can get back to us. So on this particular page, I highlighted the duration, the eligibility, the fee structure, and the various ways to adopt the scholarship. So again, a disclaimer: don't wait for the last date. Tomorrow is the last date to book the admission outrightly in case of BSc biotechnology, where test is not mandated. So it's over from my side, Madam Dalvinder.
Thank you so much, Dr. Kaur. As I can see, there are some questions from our participants. Can we take questions, Dr. Najita?
Yes, yes, you can go ahead.
Thanks for giving me chance to address your questions. And now let's start with Megha's question. Megha is asking here, what are the primary functions of blood as a liquid connective tissues?
Yes, yes. Thank you, Megha. Thank you, Dalvinder. See, liquid connective tissues, as I already discussed with you, if you're going to see in case of the RBC, see, RBC is a live cells, okay, formed elements. So the nutrition with the help of your interstitial fluid. Actually, if you're going to be see cells and tissues, which is connected with the help of your interstitial fluids one, okay. So the nutritions are transported. They are not able to transport it directly from your body. So it will take the medium. That medium is your RBC. Okay, nutrition, oxygen, it will be transported with the help of your liquid connective tissues. So liquid connective as I already discussed with you, it is playing the immune response. One more thing, lymphatic system is there. Okay, lymphatic system, we call it as a drainage system. So with the drainage system, that is also your liquid connective tissues. But what is the role of the limbs? Actually, limbs, it will be extract. We call it as a drainage system because it will be extract the excess amount of fluid. It is present in your body. It will be extracted and it will be sent to your blood flow only. Finally, it will be reach into the your like a excretory system in your kidney. The kidney filtration is takes place. After that, it will be the excretory waste are eliminated from your body. I think I can able to answer your question, Megha.
Thank you so much, Dr. Najita. Someone is asking here, what are the main components of blood and their respective roles?
Yeah. So the main components of blood, as I already discussed with you, there are two main components. One is a plasma, another one is a formed elements. The plasma, it is 55%, the liquid portion which is present in your blood, we call it as a plasma. Okay. What is the main role of the plasma? Plasma, it is playing the main role for your blood coagulation. Okay, it will be prevent from the excess blood loss while getting injury. Okay, because the fibrin is present. The fibrinogen, it is present in the inactive form, but when the blood capillaries are damaged, it will be converted with the help of your like a clotting factor into the fibrinogen. It is converted into the fibrin, that fibrin helping to clotting. So there is a plasma part. Okay, it is a watery part. 97-95% are fully water. But the formed elements, that is only living portion, that is your RBC, WBC, and then platelets. Okay.
Thank you so very much once again, Dr. Najita. I hope all the participants got the answers from our experts. As we come to the end of this webinar, I want to express my deepest gratitude to our experts for sharing their valuable insights and expertise with us. Your presence, enthusiasm, and active participation have made this experience truly special. Hopefully, the attendees found this webinar to be valuable and will be able to utilize the information for better decision making regarding your admissions. If anyone want to access this webinar again, they can visit our website, that is lpu.in, or can be accessed on YouTube also. Stay connected with us for future webinars. Until we meet again, with the permission of my seniors, I would be signing off this webinar. Take care and goodbye. Have a great day.